Now let's think about the biological library. This is a famous the bacteriophage M13. M13 has a different coat proteins, and one of them we can utilize, let's say P3 here, and we can attach those antibody-like protein expressed on here, and then utilize this as high-throughput screening system. This approach we call phage display. And now you hear movie clip to explain this concept.
This is the story about how we find antibody medicines. First we take some genes that are the molecular recipe for antibodies and combine them on a strand of DNA. We package the DNA in a protein coat made from a virus which only infects bacteria. The antibody genes make an antibody hat which is attached to the top of the virus coat. The virus is called a phage, and the combination is called a phage antibody. The phage antibody hat is unique and gives it the ability to bind to a disease target molecule. Change the antibody genes and you change the type of hat and what it can bind to. We make many of these antibody phage, and our pool has over 100 billion different kinds. Together, all these antibody phage are called a library, and this is our source for finding new antibody medicines. To target a particular disease molecule, we therefore need to find the right phage antibody from our library. For example, if we want to find an antibody to this orange one, we can use it as bait, like a fly on a fishing rod. Only the antibody phage that has a hat which can fit the orange shape will bind and stick to the target molecule. We can fish in our pool of antibody phage with our disease target and pull out each antibody phage that bites on our bait. When we find one that binds, it brings the antibody genes with it inside the coat. Because the genes are made of DNA inside a virus, we can use bacteria as hosts to make more of the virus and replicate it. We then take the genes from the antibody phage and, along with other genes, combine them to make instructions for a whole antibody medicine. Antibodies are quite complicated, so we need to ask cells to act as factories to make them for us. We can then collect the antibody medicines from the cell and use them to treat disease.
Now did you get good idea about this? Let's summarize this phage display. You can collect this DNA for the interesting proteins. Let's say this is antibody-like the proteins genome. So those genes you can incorporate into phage. Then this phage can infect the bacteria, in this case E. coli, and then they can amplify. And by breaking up this the bacteria, you can collect a lot of these virus particles, and those virus particles express this library. And then based on your the bait, this can be your target protein or target small molecule, many different things. So you can immobilize those targets on the bottom, and then only bound the virus can stay here. All others you can wash out, and the bound form you can release again, and then amplify this, and then do this cycle. So this what we call biopanning. Panning is a PAN. Original panning is to isolate out the gold from the sand. So utilizing your pan, you just shake, and then heavy the gold can sit down. All other sand is removed. That's a panning. So this is biological screening. That's why we call this biopanning. So this is the concept of phage display. Comparing to chemical library, in this case, you can utilize the genetic information of those viruses, and then once you get this virus isolated, you can amplify those by infection again, and then make more copies of those viruses, and then you can sequence them. Or even you can use RT-PCR to amplify your DNA contents, and then make more amount of the DNA, and you can sequence them. So this is very unique advantage of your utilizing biological system comparing to chemical libraries. If you look at this M13 bacteriophage again, actually bacteriophage has a different proteins. One of those very popular protein we utilize is a P3 because it has quite a long structure, so that's why we can express quite big protein like single chain FB, which has about 25 kilodalton, but copy number wise is only 5. But in comparison, on the body here, it has a lot more numbers of copies of protein, so-called P8. Now, you can express if it is a small peptide. When we use the chemical library, we expressed maybe 5 more or 6 more of a peptide on solid phase of the resin. And then each resin we need to pick it up and then sequence that by mass spec later. Maybe you can do similar things here, 5 more or 6 more, and you can express 1000 copies here and then let them bind to the target. And then select out those virus and you amplify and sequence those DNA. Then you can get those identity of your peptide sequences. So this is the power of the bacteriophages. In chemical libraries, we are talking about the millions of diversity. So one bead, one molecule approach, we are talking about millions. If it's a parallel synthesis, maybe thousands. So it's kind of order of 3 order of difference. Bacteriophage, we can make almost 10 to the 9th. It's a billion. Ribosome display or the mRNA display can be even higher diversity than phage display. In ribosome display case, the idea is utilizing mRNA, and this can be prepared from cDNA, and cDNA is representing transcriptome, and then those library can be expressed in mRNA, and then mRNA can generate protein, and by modifying those end position of mRNA, by removing stop codon, we can prevent those dissociation of mRNA after translation is over. So in this case, protein carries its genetic information mRNA together through ribosome. Then utilizing this complex, this protein can be used for screening. After this is selected, this mRNA can provide genetic information of the structure of this peptide. Then this cycle can be repeated, or this sequence can provide those information of these peptide sequences. So this is the power of ribosomal display, and this ribosome display can have maybe 1000 times higher diversity than phage displays. mRNA display is very similar to ribosome display, but a little more advanced. By incorporating puromycin, which has amino acids loaded the nucleoside the derivatives, and then once puromycin is added here, at the end of those translation, now puromycin can be transferred to the growing peptide sequence, and then ribosome can be released. Now, without this big burden of a huge size of ribosome, now only mRNA and protein can be connected, much lighter screening format. Also, this ribosome can be released. It can go for another the cycle of a synthesis of this the library member. So this can be catalytic process, much more efficient. mRNA usually is quite unstable. That's why first reverse transcription to secure those cDNA, and then screening can be done. After that, this single-strand DNA can go into the PCR cycle and either recycling of this screening or sequencing. Then this cell-free system. Right now, this one is just a cell extract and all the components for protein synthesis, and can generate a very high diversity of library source of proteins. And if you compare this with a phage display can give us maybe the billion. Now ribosome display or mRNA display even higher, 1000 times or the 10,000 times the higher diversity we can utilize. To summarize, by comparison with the chemical library we can manage those structures, but disadvantage of that was after figuring out those hit compound, and then we needed to have reasonable amount to analyze those structures. If you utilize a biological library, especially peptide library, if you can carry on those genetic information, either inside the bacteria or inside the virus, then we can just amplify those biological form. Or if it is mRNA or DNA forms, still we can amplify by PCR. That is the real strength of utilizing biological system. And utilizing this system, we can complement each other, chemical library with the biological library, to find out interesting compound.